Good morning and welcome from the National STEM Learning Centre at York. I have had several questions in from participants on the course and we're going to look at some of them now. The first question that I'm going to look at is one from Sarah, who asks, when planning a practical, is it best to do it after the explanation of the concept, or is it best to use the practical for teaching the concept? Well, Sarah, one of the things that we talked about on the course was actually having a planned outcome for your practical as to whether you were going to use it to illustrate a concept or to actually introduce a concept or to use it for developing skills within that practical. Research has shown that students very rarely are able to learn brand new difficult concepts through practical work. And if we think about the practical concept of osmosis, if you were to give a student the osmosis practical to carry out and from that deduce what the theory is, they'd find it very, very difficult to do that because they couldn't visualize it at a molecular level. So personally, if I was doing a really difficult concept such as that, I would teach the concept first and then use the practicals to illustrate the theory. I would use obviously the potatoes or the carrots experiment, and then I would possibly use the plasmolysis live experiment where you have red onion cells under the microscope and you pass over strong sugar solution and you watch the cytoplasm shrinking away from the cell wall and then passing back distilled water across the top, and you would see that the cytoplasm and the cell contents spread out to fill out the cell again. And that way you can quite easily illustrate the concepts that you have been talking about, but students still wouldn't be able to work out on a molecular level what's happening. Trina asks, what is your opinion on nudging results to practicals in the right direction? Trina, my personal view is that if we are teaching students about the scientific approach, we need also to develop within them the ability to evaluate what has gone on in our practical situation and the quality of the results that we've produced. Whether it is what they expected or not, they need to be able to try and look at the theory that they have to back it up and find out where perhaps they may have gone wrong or why their results have differed. It may be that they haven't controlled all the variables. But that's an important thing for them to develop, is the ability to actually consider are the results that they have produced accurate for what they're wanting or what they expected. If you want them to have the correct results and it's a practical that you know very often doesn't produce accurate results or doesn't produce expected results, possibly the best way forward for you is to actually demonstrate the setup of that practical and then give them some results to analyse so that they can then deduce what the patterns are and come to conclusions in that way and have your outcome for the lesson to actually exemplify that. That's what you're intending for them to learn. And the next question comes from Prashanth Good, and he asks, how can we prove the experiment with presence of oxygen in anaerobic respiration? I am assuming that what you're actually trying to do is you're trying to prove to your students that in the anaerobic setup there is no oxygen present in the test solution. Usually we test for oxygen by using relighting a glowing splint, but we can't do that in this setup, and I honestly cannot think of a way that we can test to show that there is no oxygen present in your setup. Because if you were to test the gas coming out of the delivery tube before it's collected by downward displacement, before you collect the carbon dioxide, there wouldn't be enough oxygen present in the air there to prove that to relight a glowing splint, so that wouldn't relight either. I think really that you're just going to have to tell your students that there is no oxygen there, and leave it at that. The next question comes from Hayley, and Hayley asks, how important do you think dissection is at GCSE and at A-level? And what would you suggest would be worth dissecting at each key stage? Well, Hayley, personally, I think that dissection is very important for visualising structures in situ for students. Students are often presented with a two-dimensional representation of the structures, which are simplified. And if we did not show the 3D structure and the organ actually in its situation, they would not be able to understand the complexity or the nature of actually how that structure works. Cognitive neuroscience has also shown that its engagement is really important for students' learning. And one of the parts of biology that really engages students is carrying out dissection, because they can actually see what's going on. Also, as a biologist, one of our scientific skills that we use and techniques that we've used through history is the ability to dissect out of animals and out of humans. If we think about Galen, when he was looking at the structure of veins and arteries and the circulatory system, it was through dissection that we actually were able to find out about how the system worked. And it's a really important skill for our students to learn, to be able to look at the structure and observe what's going on and link what they've learnt theoretically to the structure in place. Thank you for your questions. We hope that we see you on one of our residential courses in the near future at the Centre in York, or on one of our network courses with your local Science Learning Partnership.